Hello, and welcome to CRPS Contender, my complex regional pain syndrome education channel. This is the super simple version of You're Hot, Then You're Cold, part of my Journal Journey series. This video is talking about the paper Vasomotor Disturbances in Complex Regional Pain Syndrome, a review, written by Dr. Gunnar Wasner from the Department of Neurology and Division of Neurological Pain Research and Therapy from the University Clinic of Schleswig-Holstein in Kiel, Germany. This paper was published in Pain Medicine in 2010. Let's take an excerpt from the abstract here. Complex regional pain syndromes are characterized by blood vessel disturbances primarily affecting the microcirculation in the far part of the involved extremity. In the acute stage, inhibited sympathetic vasoconstriction and exaggerated neurogenic inflammation seem to be the major pathophysiological mechanisms inducing vasodilation. During the chronic course of the disease, as well as early in some patients, vasoconstriction dominates the clinical picture induced by changes in the microcirculation itself, such as endothelial dysfunction or vascular hyperreactivity, whereas sympathetic vasoconstrictor activity returns to normal and neurogenic inflammation is less severe. It can be suggested that the interaction between different mechanisms underlying vasomotor disturbances, as well as the severity of each single mechanism in the individual patient, have a great impact on the variety of the overall clinical picture in CRPS. And this is, again, the simple version, so I'm going to be really trying to make this very understandable, so don't be surprised if what I'm saying doesn't quite match the slide. The primary small vessels, such as arterioles, capillaries, and venules, are affected, leading to microvascular disturbances with an impact on microcirculation, skin temperature, and the clinical appearance of the limb. Skin-level expansion of blood vessels, as well as constriction of blood vessels, can be found during different stages of CRPS. There is increasing evidence that small blood vessel disturbances in CRPS are caused by multiple factors, including dysfunction of outgoing sympathetic nervous system signals, incoming pain receptor signals, and non-neural mechanisms such as endothelial dysfunction. Let's start with the sympathetic nervous system. Skin-level vascular abnormalities of the affected extremity, such as abnormal blood vessel expansion and skin warming in the acute phase, and blood vessel constriction in the later stages, are characteristic of CRPS. Studies on skin blood flow and temperature demonstrate that the warmer affected extremity in the acute stage of the disease is due to a functional inhibition of skin-level sympathetic blood vessel tightening activity, leading to skin-level blood vessel expansion. Consistently, direct measures of the neurotransmitter norepinephrine show a reduction in the affected extremity, indicating a substantial decrease of the neurotransmitter release from the sympathetic blood tightening fibers on the affected side. Norepinephrine is the primary sympathetic neurotransmitter. The loss of blood vessel tightening responses and an increased skin temperature are unlikely to be explained due to peripheral nervous system damage. It seems reasonable to conclude that a profound central inhibition somewhere in the brain or spinal cord is the underlying mechanism of the disturbed sympathetic vasocongestion in the acute stage of CRPS. Excessive sweating of the affected extremity is often found in early stages of CRPS, indicating enhanced sympathetic sweat gland activity. This opposing pattern of decreased skin-level blood vessel tightening and increased sweat gland outflow indicates a centrally located thermoregulatory dysfunction in CRPS. Alternatively, this functional deficit might also be due to autoantibodies binding to the surface of particular peripheral autonomic neurons, since 30 to 40% of CRPS patients have surface-binding autoantibodies against specific autonomic nervous system receptors. Measurements of skin temperature and blood flow over time demonstrate a change according to how long you've had CRPS. In more advanced stages of greater than 6 months, the number of patients increases in whom skin temperature and blood flow in the affected limb are decreased. Evidence that skin-level sympathetic blood tightening activity returns to normal with the duration of the disease. The initial functional inhibition of blood vessel tightening activity in the acute stage of CRPS might lead to secondary vascular hyperreactivity, either in changes where the nerve meets the target organ, which in this case is the blood vessel, or in alterations of alpha adrenoceptor sensitivity. The adrenoceptors are where noradrenaline bind to transmit signals to tell the blood vessels what to do. Such hyperreactivity could lead to increased vasoconstriction and reduced skin temperature, even though the sympathetic activity is reduced. Alpha adrenoceptor density has been reported to be increased in patients with CRPS. In addition to outgoing sympathetic innervation, pain sensory neurons can affect skin-level blood flow. Activation and sensitization of pain receptor C fibers elicit local swelling, reddening of the skin, and a spreading flare. The reddening, vasodilation, plasma extravasation, which is where fluid leaks outside of the blood vessels into the interstitial space, swelling, and pain are called neurogenic inflammation, and neurogenic inflammation might be involved in the generation of the vasomotor changes in acute CRPS. Additionally, there are non-neural mechanisms that affect the way blood flows in CRPS, in particular in chronic CRPS patients, when the cold-affected extremities in combination with trophic changes often dominate the clinical picture. Non-neurogenic vascular factors have been identified, which contribute to blood vessel tightening at this stage of the disease. At 18 months of duration, particular vasodilation transmitters were significantly reduced in the affected extremities, whereas others were intact. These particular results indicate endothelial dysfunction in chronic CRPS. Endothelial cells are the cells that line hollow body cavities, which include blood vessels. At almost 3 years' duration, there are reduced local concentration of vasodilative neuropeptides, in addition to increased amounts of vasoconstrictive neuropeptides. As peripheral vasoconstriction results in tissue hypoxia, which is where tissues don't receive enough oxygen, and acidosis, where there is too much of a low pH, followed by an increased production of free radicals, which causes a lot of oxidative stress damage, these further induce endothelial damage, which causes more damage to the blood vessels. This process might induce a vicious cycle of impaired perfusion, leading to dreaded dystrophic limbs in cold CRPS. As there is sufficient evidence that outgoing sympathetic signals, incoming pain receptor signals, and non-neural vascular factors are all involved in the vasomotor disturbances in CRPS, the question arises as to which interactions between these different mechanisms exist. In acute CRPS, it seems plausible that both the impaired sympathetic blood vessel tightening activity and the release of blood vessel expansion neuropeptides from pain receptors contribute to disturbed blood distribution. However, neurogenic inflammation alone cannot account for the blood vessel dilation and the increased skin temperature observed in acute CRPS, because intact sympathetic blood vessel tightening reflexes should be well capable of overcoming even severe reflexive vasodilation. Therefore, decreased skin-level sympathetic blood vessel tightening activity must be present in acute CRPS. Though vascular disturbances are common in CRPS, they vary significantly between individuals. In the chronic stages of CRPS, when the initially warm type turns into the cold type of the disease, multiple factors are involved, which may be mutually dependent on each other. Normalization of the sympathetic blood vessel tightening reflexes in the presence of vascular hyperreactivity leads to pronounced blood vessel tightening. Due to disease-related endothelial dysfunction, blood vessel relaxation is disturbed, resulting in even more sustained blood vessel tightening, triggering a vicious cycle of underoxygenation, too low pH, and oxidative stress damage. In very chronic cases with severe dystrophy, there is evidence for nerve fiber degeneration, so that any neural release of blood vessel expanding neuropeptides would be abolished. About 30% of patients suffer from primarily cold CRPS with decreased skin temperature and perfusion from the onset of the disease. What is the final outlook from this paper? Multiple neural and non-neural mechanisms contribute to the development and maintenance of vasomotor disturbances in CRPS. Apart from identifying so far unknown factors that may also be involved, further investigation of the interaction between these different mechanisms and its impact on the overall clinical picture seems to be of utmost importance for a better understanding and improved treatment strategies of this complex disease. It should be kept in mind that nearly all investigations have been described so far have dealt with vasomotor disturbances in the skin. However, as it is well established that CRPS particularly affects deeper tissues, further studies need to go below the surface to investigate vasomotor function in muscle and bone to further explain the mystery of CRPS. I do have 2 other videos that go further into that deeper muscle and bone one, congested capillaries and microvessel malfunction, so you might check those out. If you would like to see the paper directly or look at my notes or these slides, they are going to be linked in the description. If you like what I'm doing here and would like to help boost my content so that other people can see it and the algorithm can get more engaged, please like, comment, or subscribe. If you would like to contribute to my living expenses, my Patreon is CRPS Contender. And thank you very much to my loyal patron, Emily Malcontent. I appreciate your support. Thanks so much for watching. I hope you learned something today, and I hope to see you next time.